Hello everyone, welcome to the another video of Concept X. So today in this lecture we are going to cover the anatomy of RBC. So RBC is look like this. Suppose this is our RBC and middle portion is tapered, means concave. So if we see the basic structure of anatomy, we can see it is a biconcave, means let's suppose we cut the RBC like this way, and if we see from the side view from this section, we remove this section, we'll remove out this one, and if we see this only this upper part from the side view, this direction, from this direction, then we can see that this structure is almost look like this. It's a biconcave, means the middle portion of our RBC is tapered or it's concave from both the side, mean from the top side and from the bottom side also. The middle portion is thin and the side portion just like a ring, the side portion it is thick and the middle portion is thin. So here it is the thin portion, that middle one, it is very much thin, and the surroundings is thick. From the side view, our RBC is almost look like this. It is a section view, means we have removed one section and this is the another half, and from the another half, and in this half if we see from the side, then our RBC is look like this one, that the side portion is thick and the middle portion is very much thin. So that's why it is called biconcave. Biconcave means from both the side of the middle portion is concave, means bent, narrower, or you can say it is a discoidal. It is a disc-like structure. That's why it is called discoidal. What is the diameter of our RBC? Diameter is 7 to 8 micrometer. Diameter means from here to here, this much of range. It is the diameter of our RBC. So diameter is 7 to 8 micrometer. Then another thing is the plasma membrane is very much strong and it's flexible. So one of the major benefit is when our RBCs are continuously traveling throughout our capillaries. So a few capillaries, they could be very much small. When one big artery is converted to arteriole, then this RBC may adjust their body shape. So for the adjustability, this kind of flexible and strong nature will help our RBC to go through a narrow artery. When a big artery is converted to small arterioles and then capillaries, then same RBC, it will help to go through that narrow arteriole without any kind of problem or without any kind of difficulties. So that's why the strong and flexible nature of our RBCs, it helps to move through the narrow or small capillaries. Inside of our RBC, there is no nucleus. No nucleus is present, and other cell organelles are also absent, means mitochondria, ribosome, nothing will be there. Nucleus is also absent. So this is the another characteristics. So RBC cannot reproduce. If nucleus is not there, then chromosome, chromatids, nothing, sister chromatids, nothing will be there. So our RBC, it cannot reproduce. So what is the average lifespan? It's 120 days. And after 120 days, it will go to spleen. It will go to spleen, which is a graveyard. Of RBC. The spleen is a graveyard of our RBCs. So after 120 days of their circulation inside the blood vessels, our RBC will definitely go to our spleen and it will got destructed. So this RBC they form inside the red bone marrows. So what is red bone marrows? For example, suppose this is one bone. So our red bone marrow is this part. This is our red bone marrow, and this is our yellow bone marrow. And here again we have some part of red bone marrows. So this is our red bone marrows, and this is our yellow. I don't have any yellow colors, that's why I'm drawing it with orange. So this is the red bone marrows. This is red bone marrow. This is also red bone marrow. So our RBC, they form inside these red bone marrows, and then they will go to the blood circulation. So the thing is, if there is no nucleus and no cell organelle is there, then from where these kind of hemoglobin molecules, those are present inside our RBCs. As we know that our RBCs, the main purpose of RBC is to transport the hemoglobins, because hemoglobins carry those oxygen molecules. And when RBC travel across the blood vessels, then hemoglobin, they are present inside those RBCs. So RBCs is nothing but a carrier molecule for our, or carrier cell for our oxygen carrying hemoglobins. So these green dots, they are nothing but the hemoglobins. So these are hemoglobins. So these are hemoglobins. So these hemoglobins are found inside the RBCs. So if the question arises, if no nucleus and no cell organelles are there, then from where these hemoglobins are going to be formed? There must be some kind of precursor molecule. So thing is, before the formation of a mature RBC, our RBCs will be considered as reticuloblast, and the main precursor cell of our RBC is erythroblast. Erythroblast is basically large in size. So when erythroblast is converted to reticuloblast, when erythroblast is, suppose this erythroblast is first converted to reticuloblast, and these reticuloblast will convert it to RBC or erythrocyte. So in reticuloblast, our hemoglobins are not found. There is no hemoglobin as there. But when erythroblast is slowly converted to reticuloblast, then few hemoglobin molecules are start to form. And when this reticuloblast again converted to erythroblast, during that process, most of the hemoglobins forms. So in that case, in erythroblast, when nucleus is present and other cell organelles are also present, what happens? When this erythroblast is converted to reticuloblast, our nucleus and other cell organelles are start disappearing, and slowly hemoglobin are start to form. So in erythroblast, nucleus, other cell organelles are present, for example. So, but no hemoglobins are there. So when erythroblast converted to reticuloblast, nucleus along with other cell organelles start to disappear, but appearance of hemoglobins are about to start. So slowly in reticuloblast, cell organelles are disappearing and hemoglobins are appearing. So hemoglobins molecules are start to form inside the reticuloblast. when the reticuloblast is converted to mature erythrocyte, during that transition, what will happen? More and more hemoglobin start to form, and complete disappearance of nucleus and other cell organelles are there. So from erythroblast to mature RBCs, cell organelles are disappearing and hemoglobins are appearing. In erythroblast, hemoglobins is less, but in erythrocyte, hemoglobins is more. In erythroblast, cell organelles are there, but in RBCs or mature erythrocytes, cell organelles are not there, not present. And they are formed inside these bone marrows. This is called the epiphysis. This portion is called epiphysis. And this portion is called diaphysis. So this is the diaphysis part, and this is called the epiphysis part. This is the epiphysis. This is epiphysis, and this is diaphysis. The middle portion is diaphysis, and end parts are called epiphysis. So in epiphysis, red bone marrows are there. So inside the red bone marrows, our RBCs are formed. And what is the structure in RBCs? I have given all the information. This is the biconcave in nature, discoidal structure, 7 to 8 diameter, no cell organelles, nucleus is nothing is present. So those hemoglobin, those are formed inside that erythroblast. They are basically from our reticuloblast. So this initiation, starting of formation of hemoglobin, it is formed inside the reticuloblast. This stage is started in our reticuloblast, which is the intermediate precursor molecule of our RBCs. So main precursor is erythroblast, then erythroblast converted to reticuloblast, then reticuloblast is converted to mature RBCs or erythrocytes. So in erythroblast, no hemoglobin is there. In reticuloblast, slowly hemoglobins are forming, and in mature erythrocyte, it is nothing but a pocket of our hemoglobins, where all the cell organelles are already disappeared. And this everything is going on inside this red bone marrow. And from red bone marrow, when this mature RBCs is formed, they directly come to the blood circulation. So this is the overall. And after the 120 days, from 120 days, where will go our RBCs? RBC will go directly to our spleen, and where it's a graveyard, which is also called as graveyard of our RBCs. And inside that graveyard or inside that spleen, our RBC will be destroyed. And those hemoglobin molecules, it will directly send back to the liver. So hemoglobins are destructed inside that liver, and that RBC cell is destructed inside our spleen. So this is the overall information of anatomy of our RBC. So hope you like this video. Thank you.